question, Francisco. I enjoyed very much your talk. So do all human ALS patients have this phenotype, the TDP43 aggregates?
No, obviously the ones that have the mutation of SOD do not. All the others, the C9 mutation, sporadic cases, FUS mutations, all of them have TDP43 inclusions. That's why we think that we should worry about the inclusions, because whatever the cause that is producing them, we can mitigate the problem by reducing them.
And what about the gene encoding the TDP43?
Yes.
The gene in patients and in normal people is the same?
About 5% of the patients present mutations.
Okay.
The mutations are functional. So you take the mutations, clone them, do splicing assays or things like that, and work fine. Probably they predispose to aggregation. That's the only guess that we can do. But most of them are in the C-terminal region of the protein.
Okay, thank you.
Hi. I enjoy very much your talk.
Thank
the
you.
antidepressive medications, the pills, they will block or delay the aggregation, or how this antidepressive
when we use the drugs?
Yes, anti-stress, and regular, but they
The
will...
stress, we don't know. That's why it's maybe about what produces the stress, we don't know. What we do in our mechanism is provide a high local concentration of the prion-like domain, and then aggregates. Then the resolution of the aggregates apparently goes by some protein degradation pathway I was discussing yesterday. These products that we use stimulate autophagosome formation. And they do, but the protein degradation seems not to go completely through there, because if we inhibit the fusion of the autophagosome with the lysosome, nothing happens. They clear all the same. But if you inhibit the proteasome, they don't clear anymore.
Very nice talk. Thank you very much. I have two questions. One is, what is the function, the physiological relevance of the decay in the concentration of TDP, and what happens if you, for example, overexpress in an adult animal the TDP? Is there a pathology associated with it?
Not actually, no. You can overexpress, in flies at least. In mouse as well, there are several transgenes of mouse that if you do too much, then yes, it's toxic.
Yeah.
If you instead a level of twice as much of the endogenous, they're okay. I don't know what is the relevance.
So if there is no decay, not necessarily that leads to pathology?
No, the conjunction of the decay with the aggregation is what leads to pathology. The decay is fine.
Wow.
I mean, the decay, obviously, whatever TDP43 is made in brain or liver is enough in normal situations. But if that protein is being catched by the aggregates, then it's not.
And the second question, quickly, is it known where is, at what phase of the protein life is the aggregation occurring? Is co-translational, post-translational?
No, it is not. It can happen both in the nucleus and in the cytoplasm. So I think that it's post-translational, and when it's shuttling, something may happen, probably some misfolding of any kind, that at times is clear immediately, and at times for some reason, maybe decay of the clearance system or something like that is not clear, and it gets formation. What we don't know is in the patients. I mean, we see in the patients the end point. How many people has inclusions in the brain and goes around happily is difficult to tell. There is no tracer still to see it in vivo. Pablo.
Stephen McKnight has described aggregations of proteins without membrane that I don't know if they're amyloid-like. Is this something, an example of this, or different?
Okay, we cannot stain these aggregates that are a model with for amyloid or for amyloid fibrils. You know, the what is the staining is well, the staining for amyloid is negative. The other theory is that there are stress granules that then grow. And then you look for TIA, that is the one of the markers for stress granules, and we don't find it either. So we don't know. We don't know. I don't know if it has a membrane or not, really. Yeah.
Did you try to mitigate accumulation of aggregates with rapamycin or other means of inducing autophagy?
Sorry, I lost your question.
If you try to mitigate the accumulation of aggregates with rapamycin or other means of inducing autophagy.
Yes, yes. Rapamycin helps, not as efficiently as the tricyclic antidepressive. So it does reduce the aggregation.